I welcome everyone to this live webinar session of the Research Viewpoint by Karger. And the topic of today is the exosomes and microRNA, a new form of neuroimmunomodulation. This is the blog for your reference, and that's also where the recording of the webinar will be posted afterwards. So a few housekeeping rules. First, I'm Teresa Matthews, publication manager for Karger. I am responsible for the neurology portfolio, and I'm also publication manager for the journal Neuroimmunomodulation. I just wanted to let everybody know, if you do have questions, to use the questions box or the chat. We will do the best that we can to monitor those two channels. As I mentioned before, the webinar will be recorded, and the access to the recording will be given via the Research Viewpoints blog afterwards. And feel free to use the chat for reactions and comments, but please keep respectful and on the topic. The agenda for today is this welcome and introductions. Then we will have one lecture of 20 minutes by Professor Monika Fleshner and another lecture by Professor Dr. Rainer Straub, also 20 minutes. And then we will have time for questions and answers and any closing remarks at the end. So I would like to introduce our guest, Professor Dr. Monika Fleshner, Professor of Integrative Physiology from the University of Colorado, Boulder in the US, and Professor Dr. Rainer Straub, Professor of Experimental Medicine from the University Hospital in Regensburg, Germany, and also the editor-in-chief of our journal Neuroimmunomodulation. Monika, you are the first one to start.
Great. Very, very nice. Thank you. So thank you everyone for joining us in our little brief lectures today. And I want to tell you a little bit about exosomes, danger-associated molecular patterns, or DAMPs, and microRNA, a new form of neuroimmune communication. Next. So I've spent the majority of my career trying to understand the stress response. And the stress response can be envisioned or seen in every aspect of life, as well as molecularly. So there are molecules that get impacted by challenges in their environment, and we have evolved ways of trying to protect those molecules, as well as protect the cells, and as well as protect the whole organism. Typically, when people think of the stress response, they think of the stress response to a whole organism. And I'd like to remind everyone that the stress response is both friend and foe. Next. So the acute biological stress response promotes adaptation and survival. So we as humans, as well as other animals on the planet, have survived because of the complicated and beautifully integrated stress response. Next. Typically, we think of the stress response as the means by which we fuel the fight or flight response. So if there's a challenge in our environment, the stress response gets kicked in that allows us to breathe more heavily, dilate our pupils, our heart rate begins to pound. We have a cascade of endocrine responses. We know a great deal about this response. You have the autonomic nervous system branch, which is comprised of both sympathetic and parasympathetic neurons that have their origins in the central nervous system and then branch out to many tissues in the periphery. Once again, all of these responses are designed to fuel success in the fight or flight response. Next. More recently, though, the field has recognized that beyond simply sympathetic nervous system responses that, have our heart rate start to go up and dump glucocorticoids, HPA responses, so that our muscles have blood and we have energy and we can run. The immune system also should be a part of this orchestrated stress response. And so the acute biological stress response also includes a very beneficial effect to the immune system itself, so that we actually, by turning on the acute stress response, can fight and kill bacterial challenges better. We can recover from a wound more quickly. All of this is a part of this friend aspect of the acute biological stress response. Next. Interestingly, the stress response includes changes in the inflammatory system, so in the innate immune response. And we have a process that exists that responds to damage, for example, and that's often referred to as the local sterile inflammatory response. So an example would be if I took a hammer and slammed it on your thumb, you would have changes in that tissue that include the release of cytokines and different kinds of proteins that are designed to promote inflammation, allow for cells of the immune system to migrate into that tissue. And in the end, that's a great thing to happen. So your thumb recovers and your swelling goes down. Next. However, the beneficial effects of this local sterile inflammatory response, as well as the systemic sterile inflammatory response, which is when this response goes beyond the local to the entire body, is context dependent. Next. So what I mean by that is I just gave you an example of the hammer on the thumb, and that can be a very adaptive thing, and your thumb will get better. But you can also suffer maladaptive consequences of both local sterile inflammation as well as systemic sterile inflammation. That maladaptive aspect, for example, if you yourself suffer from a disease that has an inflammatory component, like multiple sclerosis, for example, or even some of the neurodegenerative processes, you turn on this sterile inflammatory response, and that will interact with that disease or pathogen, that pathological process, and make your symptoms worse. And in fact, the systemic sterile inflammatory response can even be fatal. So here you've got a process where the immune system is responding not to pathogens, so not to bacteria or viruses, but rather to damage or a threat. And we understand quite a bit about the signaling that's involved with that process. Next. So here is a little picture. What you should be envisioning is that you're looking at the edge of a cell, some type of an inflammatory cell. This could be a macrophage if it's out in the periphery, or it could be microglia if it were in the brain. And I've told you that the sterile inflammatory response can trigger these cells to respond in the absence of a pathogen. One particular kind of signal is a danger-associated molecular pattern, or a DAMP. One that I've spent quite a bit of time studying is an intracellular chaperone called heat shock protein 72. So this protein, this molecule, upregulates when a cell is under challenge. It got its name from a series of experiments where scientists demonstrated that if you heat fruit flies up to a temperature over and over again, they can survive a higher temperature with that prior experience of being exposed to heat. And one of the molecules that helps do that is this heat shock protein 72. So when this molecule's inside the cell, it's extremely adaptive. But once it's outside of the cell, it acts as a trigger to activate inflammatory cells. So that's why that's called an E, extracellular heat shock protein 72. And we know a lot about the signaling that's involved with that. Those DAMPs can bind to receptors, and they can be found floating freely in the blood, but they can also be found in these vesicles that we're going to talk about today. The other component of that signaling is the microRNA, and the microRNA become packaged with both the stimulatory damps and the microRNA themselves. Next. So here's a brief introduction on exosomes, damps, and microRNA. So the process by which we end up with these small extracellular vesicles occurs via the initiation of an endosome. There's a great deal of detail understood about how this all works, where you have these little blebs of vesicles that are in the multivesicular body. And then with the cell, one of the primary signals is an increase in intracellular calcium, which then allows this cell to release these little bubbles. Next. And what's fascinating about these small vesicles is they contain a lot of information, a lot of signaling information on their surface. People who studied these microvesicles have struggled for a very long time to try to understand, how do we know when we're looking at a vesicle that we're going to call an exosome versus other kinds of microvesicles that are also found out in the blood. So an exosome has certain features. It expresses certain proteins in the membranes. And if we look inside next, oh, I guess I didn't do anything. They possess both information on the inside, so intracellular cargo, as well as information on the outside, their cell membrane wall. Now, why would the body choose, in a situation where you're going to trigger a sterile inflammatory response, to package these important signals in an exosome versus just releasing it out into the blood? People at first thought that these little vesicles were just carrying garbage out from the cell, but there's been a great deal of research trying to understand the unique sorting and packaging of the intracellular and the intravesicular cargo. Maybe these little vesicles are a way that we can do novel cell-to-cell signaling and potentially long-distance communication. So typically, cells of the immune system sit side by side. Many times their signaling requires cogent interaction, so they literally have to touch. But here we've got a situation where we've got these signals that we know are immunostimulatory and immunoinhibitory, and they're getting packaged in a little vesicle and are able to float around from perhaps periphery to the brain, or maybe from the brain to the periphery. Next. This delivery system could be targeted as well. So one of the molecules that are in the membrane of these vesicles are these intracellular adhesion molecules, which are zip codes for these little vesicles that then bind the appropriate receptor on the distant cell. The other thing that's cool about packaging these immunomodulatory signals is you can have a complicated signal, if you will, both stimulatory and inhibitory. And then furthermore, by having this lipid bilayer, you can protect the intraexosomal products themselves, the contents, from degradation. Next. Next. There. So how do you go about trying to do these sorts of experiments? So let's say you're very interested in exosomes and you're interested in identifying these little vesicles that can be found, by the way, in almost every liquid in your body. You can find exosomes in breast milk, in saliva, in urine, as well as in the blood. And so you need to do a particular type of isolation method. And this is typically required. You need to be able to visualize these small vesicles. You need to be able to demonstrate their size. You need to be able to demonstrate that they express on their membranes these classic exosomal markers, CD63, RAB5B, for example. And then you can look for the intracellular cargo. So transmission electron microscopy is sort of the gold standard. Next. And so in our experiments, we were able to demonstrate that these little vesicles that we believe we are isolating and enriching our plasma fraction with these vesicles We see that they're the right size. If you look in this image, this is a TM image, and you can see that we have nice round vesicles that are about 80 nanometers in size. Next. The other things that we needed to do is we used an instrument called Nanosite that is a particle counter, and it also judges the size of that particle. And what you can see, can you see my pointer? No? Yes? Do you guys see a pointer? No? Okay. So anyway, what you can see is in the black peak here, that is particles that are in the enriched fraction, and they're just about that golden size, 74 nanometers. Whereas in the depleted sample, you can see the different axis here. We've pretty much cleared out all of those vesicles. Next. And then the next verification or validation that you're really studying exosomes is you look for these markers of an exosome. And so this is an exo-ELISA. It's a way of measuring the expression of CD63 in the exosome-enriched fraction. So these vesicles are expressing this marker, as well as here we've got a double marker, both CD63 and RAB5B. So we're pretty confident in our enrichment process that we have exosomes. Next. So what we know is that exposure to an acute stressor, so if you were to take a preclinical model of acute stress, next, stress increases the expression of this heat shock protein 72 on CD63 positive particles, so on exosomes. Next. We then wanted to demonstrate isolation of these CD63 particles, and we used a magnetic bead that has a protein on the surface. We stick an antibody that binds this CD63, and that allows us to pull these vesicles out of our solution if we then wanted to validate what was in those vesicles. Next. And what you can see is that the exosome-enriched samples, indeed, we can pull out the CD63 positive samples here. Next. And we demonstrate this is the heat shock protein that we're hypothesizing is being expressed on this vesicle from a stressed animal. And we can demonstrate that, yes, indeed, it's on a CD63 positive particle, because if we pull those particles out, you could see the signal for heat shock protein 72 goes down. So these are the types of studies we have been doing to demonstrate exposure to an acute stressor does increase expression of this very immunopotent DAMP, HSP72. Next. But today's focus is also on microRNA. And so we have the positive immunostimulatory signal from a stressed animal, which is the expression of a DAMP. But what's fascinating is that within these vesicles, we also have microRNA. And this particular panel, we measured 68 microRNA proven to be involved in modulating or regulating the immune system. And you can see we've split this into two different sides because some of the exosomes have very high concentrations of microRNA and some have very low. And in order to see what's happening here, that's why we've got a graph this way. And not to belabor each individual microRNA, but fundamentally the majority of the microRNA that changed, they were reduced. In addition, what we can show is that there's a relationship between the microRNA and the IL-1 beta, so one of the most potent cytokines in the blood. What do microRNA do? So they do lots of things, okay? They're classic suppressors. So typically, if we see a reduction in a microRNA that's involved with the immune system, you would predict that you're releasing the immune system from inhibition. So tonic inhibition would be a stable level of these microRNA circulating through the body, perhaps through the brain, and trying to keep the entire system quiet. But then when you get exposed to this acute intense stressor, one of the ways to really turn on the immune system, that powerful inflammatory response that can be adaptive, is to turn up the DAMP and turn down the the microRNA or the break. And what's interesting as a stress physiologist to me is this process seems perfectly logical. It's the same sort of thing we see with sympathetic and parasympathetic regulation. So during a time of stress, you activate the sympathetic nervous system, so your heart beats very fast, and you withdraw parasympathetic inhibition. So it's the combination that really allows the body to have this potent inflammatory response to an acute stressor. Next. Now, of course, as is always true of science, it's very complicated. What we've learned is that these microRNA rarely function in isolation. They tend to function in teams. And we've really tried to figure out creative ways to understand in the whole circulation what's happening during an exposure to this acute intense stressor. And so do you have some microRNA going up and others going down? Do you have some microRNA staying the same and others going down? And so one way we just started to tackle this question is using a type of analysis called K clustering. And what K clustering allows you to do is allows you to take all of these different microRNA that we assessed in the exosomes that were purified from the plasma of a stressed animal and put them all together and see how they cluster together. And what this image is showing you is the different microRNA that comprise this first cluster on the left, and the second cluster, third cluster, and fourth cluster. So you can see these are the members of that cluster. The blue arrow is associated with that cluster. And so we're trying to understand, can we learn something about this dance that's happening in the circulation during exposure to an acute stressor, rather than trying to drill down to a single microRNA. Next. So the big picture conclusions that I tried to share with you today are the following. Inflammatory homeostasis likely involves exosomes, danger-associated molecular patterns, and microRNA. So high levels of these repressive microRNA from non-stressed organisms, for example, can constrain inflammation. So we've demonstrated that by taking exosomes from a non-stressed animal that have higher levels of microRNA and low levels of DAMP, and put them on cells, on macrophages in culture that have been stimulated to make pro-inflammatory cytokines. And when you do that, those exosomes quiet the inflammatory response. So that's a fascinating idea, and that process is being pursued primarily from exosomes from, for example, stem cells or things like that. Stress triggers systemic inflammation by reducing the cargo, the exosomal microRNA in the vesicle, and increasing the DAMP expression. Next. And so this concept of immune homeostasis is depicted in this image here. And what you can envision is a tonic inhibition of the peripheral inflammatory response. During exposure to a stressor, you increase the DAMP and you decrease the microRNA. We know the sympathetic nervous system plays an important role in increasing DAMP expression. So these changes that are happening with the immune system are a part of the acute stress response. And that can be highly adaptive under normal circumstances. However, if this response is driven over and over and over again, or if you were exposed to an extremely traumatic stressor, this can lead to problems that are associated, health-related, both mental and physical problems that are health-related, associated with chronic drive of this powerful system. Next. So there's a great interest in exosomes as biomarkers. You can see here, these are all reviews from the last year. But there should be some caution put out there, because actually, if you look at these titles, it's problematic to try to find a single, definitive biomarker that's associated with a pathological state. Here we're looking at the concept of surveying exosomal microRNA as a way of a biomarker for brain inflammation. And you can see this, there's some successes and there's some failures. In addition, I want to point out that although I primarily looked at microRNA that we know are inhibitory to the inflammatory response, some of these microRNA go the other way. So you can have both directions. Next. And then lastly, one of the other reasons I think understanding the impact that stressors have on these exosomal cargo as well as their membrane protein expression is that as we continue to pursue biomarkers from analyzing exosomes, we need to consider the state of the human during that blood draw, where you're trying to determine, for example, has their cancer come back? And for many cancers, the percentage of relapse is frightening. Glioblastoma, 89% relapse, and so forth. And so you've got this person that's in your experiment, they go in, they're very scared and nervous, and you draw blood, and it's very possible the stress of that situation is sufficient to change the microRNA cargo and membrane expression. In fact, if we look here, there are many biomarkers that are reported in the literature to be cancer biomarkers, and a great many of them are actually stress responsive. Next. So I want to encourage everybody in the audience to continue this type of research. I think it's a fascinating field, but as is always true in science, when you start working on a topic, it seems straightforward, and then the longer you work on that topic, the more complicated it becomes. And so one of the things people need to be very careful about is understanding which microvesicle they're looking at. If they really want to say they're looking at exosomes, they need to be very careful on validating the expression of the well-known biomarkers, the protein markers for exosomes, as well as being careful when they're measuring the cargo that they actually are capable of rupturing these little vesicles. Next. So we have a lot of work to do on this topic. There's my kids waving to you. I want to acknowledge Tel Kelly. This is his primary dissertation research, and Shelby Hopkins they work together on almost every experiment. And Camilla Crane was one of the graduate students several years ago that started some of this work. And that's all I have, and I'm excited to hear Rainier's, Dr. Straub's talk next. And I look forward to answering questions if there are any.
Thank you. Perfect.
Thank you. So we will go to Rainer's presentation, and then we will have the questions and the answers at the end. So, Rainer, you're up.
Yeah. Monika Fleshner had some important experience in this work, but my experience is relatively little. I'm looking more as an editor-in-chief on the thing, and what we saw in recent years was that there were many papers submitted to the journal, and the papers often did not really touch on exactly the idea of neuroimmunomodulation, and what I want to show here is what I would think is important for these microRNA studies to be eligible in the neuroimmunomodulation field. Next slide. Teresa, next. It starts, all the thinking starts somewhere around energy. We got this energy from the sun, and we produce, we got the different elements, and in the end we produce the ATP, which I would call the coin of energy. Next. Two organs could be called selfish, the brain and the immune system. When it comes to certain threats in nature, like the fight and flight, thirst, cold, heat, starvation, hemorrhage, then the selfish brain has the hierarchically highest level of control. When it comes to the wounding and infection, then it's the immune system, and they fight about this energy coin, the ATP. And most other organs under normal conditions are hierarchically under these two important organs. And to give you an idea how much extra energy would be needed, I have listed some important things. Next, Teresa. For example, inflammation, there the additional costs relative to the total costs under the healthy conditions is 25 to 60%. Even in chronic smoldering infection is 10%. With acute pain is up to 60%. With chronic pain, 15%. Psychological stress, 30%. Sleep alterations up to 30%, and anxiety 10%. Smoking makes an extra 15% if you are doing that. Of course, this list is not complete, and some more can be found in the literature. The literature to these numbers are given in the mentioned literature here. From this point of view, the two organs must work with each other. They must talk to each other. Next slide. And what we recognized over many, many years now is that typically we mention here the hormonal aspects through the endocrine glands, most often the HPA axis, hypothalamus, pituitary adrenal axis, but also the nerve fibers, which was called hardwiring, nerve fibers that connect the brain and the immune system. But the question is next, whether there is an additional pathway that can help. Next slide. Yes, the additional pathway. And next slide. With the idea that Monika brought up already in the years 2013 and 2014, that stressful events can release not only the DAMP that she talked about, heat shock protein 72, but also some important microRNAs. In her situation, she saw the decreasing of microRNA 142, 5p, and 203, but sometimes you will see this a little later, the things can go the opposite way. Two next slides, because I don't talk about this because Moni has already mentioned this. I go to the next one. In the preparation phase of making this webinar and the entire invitation to the audience, I made a literature survey of more than 5,000 references included, and I included into the paper 798 references. I was lucky because in neuroimmunomodulation there is no limit as to the references. And my questions at the beginning were, are there microRNAs from brain regions that can influence immune function? Are there microRNAs from the pituitary adrenal gland that can influence immune function? Are microRNAs secreted during the stressful events? That is what Moni already showed in her presentation, but there are some more. And then from the other side, from the immune side, from the immune system side, do microRNAs from the macrophages, different types, or dendritic cells, influence neural function or microglia? The same for the neutrophils and monocytes, the same for different types of T cells and of B cells. And in the end, I had 12 tables with a very heterogeneous picture, but nevertheless, it gives an idea what's happening. And it's not easy to summarize all this information into one slide, but I try to do this. Next slide. Here I show those publications that reported anti-inflammatory and pro-inflammatory effects on immune function. And on the x-axis, you can see the different places in the brain or glands, pituitary and adrenal. And on the y-axis, you see the number of references supporting one or the other direction. And it's really funny. Most of the microRNAs that are secreted from the hypothalamus, if you take them and look, and many people have looked on these microRNAs with the functions on immune cells, but not in the same paper, in different papers, they saw that most of these microRNAs from the hypothalamus are anti-inflammatory. And very similar is also the situation looking on adrenal glands. So most of the microRNAs released from adrenal gland cells are anti-inflammatory. Pituitary gland with a similar picture. Not much is known at present for amygdala, the sympathetic outflow or the parasympathetic outflow. All in all, 168 papers showed anti-inflammatory microRNA and 38 pro-inflammatory microRNA. Next slide. Now we come to the question of stress. And the different types of stress that are applied either to animals or to humans are given on the x-axis. And in the same way, the number of references supporting the anti or pro-inflammatory role of microRNAs on the y-axis. And you can see that different types of stress can produce microRNA that is anti-inflammatory, particularly looking at social isolation, the chronic social defeat stress, or the Trier social stress test in humans. That's number 3 from the right side. That is, most of the papers showed microRNA that is really anti-inflammatory, but with respect to sleep deprivation, it's the opposite. And all in all, this is a really heterogeneous picture here with the stress. 136 showed anti-inflammatory, showed microRNA with anti-inflammatory effects, and 110 had microRNA released with pro-inflammatory effects. The next slide gives now the opposite direction from the immune system to the brain. Here, it's interesting that the M2 macrophages, we usually would define as anti-inflammatory pathways. They had nearly all of them had produced positive or protective effects on neural function and neural structures. That's also true for the dendritic cells or for the T cells in general. The B cell also had most microRNAs that are released from the B cell had protective effects in the brain, but it's also of course a heterogeneous picture. But from this point of view, next slide, we can clearly say that there might be, and is highly likely, that immune cells can release some exosomes with microRNA that can enter the brain, or the other way around, that they can probably talk to each other using this pathway. And at the end of my talk, I just want to show you an example how a paper can be produced that very nicely shows the connection. Next slide. In this experiment of Young and colleagues that has been published in Brain Behavior and Immunity this year in 2025, they focused on psoriasis vulgaris in animals. Next slide. They used a technique, it's called imiquimod induced psoriasis-like disease. And they first stressed the animals, and then they gave imiquimod for 7 days. They used chronic social defeat stress, a well-known stress paradigm. And what they saw, which is just shown here on the right side of the slide, is that these animals, after the stress, had more anxiety and more what we would call a depressive-like state after the stress. That has nothing to do with the imiquimod. But on the next slide, you see the links then to the disease. Those stressed had more epidermal ridges, which is a sign for the psoriasis development. And what they also do, they had more scratching, at least on day 7. They started the scratching with this imiquimod, and they had it more if they were stressed. Next slide. And they also have demonstrated that with the imiquimod and with the appearance of the disease, there are more CD68 positive macrophages in the psoriatic skin. This is the green color on the panel A. On the right side, in the psoriatic skin, there are more of these green little dots, and also the thickness. You can see that also on this image. The thickness is higher, and the number of epidermal ridges, which is this marker of psoriatic skin disease, is also higher in the psoriatic skin. And if they use the glutronate, glutronate is a tool to remove macrophages in the periphery, then it can be clearly seen that those animals with the stress and imiquimod and psoriasis that usually have the high numbers of macrophages, with the glutronate they have the lower numbers of macrophages, and they have the lower thickness of the epidermis, and also the ridges were much lower. Next slide. Now what they also found, they found that nerve fibers seem to play a big role. The nerve fibers are demonstrated as positively stained with TRPV1, which is the capsaicin receptor on sensory nerve fibers in red on the left side in the panel D. Those nerve fibers were in close contact to the macrophages in green, CD68 positive macrophages. And when they use this resiniferatoxin, which is a potent agonist that destroys the sensory nerve fibers after 3 days, then the macrophages were gone in the model with the stress and the imiquimod, which is panel E. And then also the score changed of the disease score, the scales or the erythema, both were lower by removing the sensory nerve fibers. And next slide. Now they linked this interesting thing with the microRNA LED7B, and they looked into the dorsal root ganglion, and they knew now that the sensory nerve fibers might play a role. They looked into the dorsal root ganglia, and they have seen that the microRNA level in the stressed animals from the dorsal root ganglia is clearly increased on day 0 and also a little on day 7. And in addition, they saw on A, they saw the expression, and B, they saw the release. And then they blocked the LED7B microRNA in the dorsal root ganglion by injecting a blocker into the root ganglion in the animals with the stress and the imiquimod, and they saw that the blocker clearly reduced the scores, the scratching number, the epidermal thickness, and the epidermal ridges. And in further experiments, I don't show that in all the details, it's their work, or can be easily seen there in the brain behavior and immunity. Further experiments showed that the effect is dependent on toll-like receptor 7 positive macrophages. They used an animal model with a knockout because the microRNA from the sensory nerve fibers bind to the TLR7, and this receptor on the surface is a dump receptor. As Monika showed, they play an important role in the immediate reactions. And the microRNA LED7B seems to be a clear signal now from the brain through sensory nerve endings into the tissue to induce a macrophage activation in the tissue. And this clearly shows the link between the brain and the immune system in an animal model of chronic inflammation. Next slide. So what we can see here, an important experiment that can show the connections between one and the other system would look like this. It would be nice to see how the microRNA source exists in the brain, finding a defined brain structure or nerve endings or endocrine glands that produce upon the brain stimulation the respective microRNA. And then we need to check the peripheral presence of this microRNA in the body fluids or around the nerve fibers, as has been shown in this example. For example, in plasma, nerve terminals, neural ganglia, in other body fluids, or released even from the endocrine glands. And then to check whether this microRNA can influence directly the immune function, for example, cytokines, chemokines, migration, immune tolerance in inflammatory disease. That would mean an in vivo experiment, like in the last example. And the other way around, next slide. Here we ask, we ask ourselves, go back one. Teresa, I want to finish this little thing here. We want to find the microRNA, the source in the immune cell. We need to find the immune cells. We need to find the stimulus that would be necessary to release also the microRNA, and the location in the immune cells. Where are they? Are they in the spleen, the lymph nodes, or elsewhere? And to check whether they are released into the blood or plasma or in lymphoid structure or other body fluids, and whether then they change neuronal function. For example, locally in a tissue, it could be the release of neurotransmitters or peptides, but it could also be a direct influence on neural function, even behavior, or memory functions, or pain gating, pain entering into the brain. So next slide. I think I finish here. What I want to stimulate now from the audience is that with these ideas, one can nicely demonstrate neuroimmunomodulation, and this way or this way. And these are interesting papers that we would be happy to see in the next weeks or months or years. Thank you for your attention. Now we open for questions.
So yes, Rainer, thank you so much. Thank you so much, Monika. And we do have some time for questions. Let me just check here. Yes, I have a question for Monika. Monika, can one identify cellular type and cellular state based on exome profile? Is this possible?
Yeah, so that's a wonderful question, and that's precisely what we have been trying to do. So for example, some kinds of, if we want to stay focused on brain, when you have a closed head injury, like a mild traumatic brain injury, one of the questions that are often asked is do we have any idea what part of the brain is disturbed? And so if you have a cellular stress response in the brain, you would be able to tell that, and you have a neuronal signal. So you have a protein that signifies that this exosome came from a neuron. If it has this change in these stress proteins, you can conclude that that cell that released it was in a state of cellular stress. Yes, precisely. And that's all I have.
Thank you. Thank you, Monika. Do you have any comments on that, Rainer? Is there anything you wanted to compliment, or can I move to the next question?
We'll go to the next question.
Yes, okay. I do have a question for you. You showed that the crosstalk between brain and the immune system serves energy control of the entire body. And the question is, are the microRNAs that control energy regulation coming from the brain or coming from the brain and the immune system?
Well, that's a good question. Interestingly, this has, similarly to the demonstration that I presented before, not demonstrated in one single big experiment where you really define the place in the brain where the microRNA is released, and then how it can, for example, influence insulin resistance, or which is an important part in energy regulation. Or another way would be uptake of nutrients in the gut. That would mean you find a microRNA that is released somewhere in the brain and travels through an exosome into the gut and influences there, for example, uptake of triglycerides from the gut. Things like this had never been shown, and that would be also highly interesting. What has been shown is that there are single elements, microRNA elements, that can influence these different parts, but in one single experiment where you see the in vivo aspect, and you clearly define the location in the brain and in the periphery. This is the thing that is so important and not much demonstrated.
So can I ask, so Rainer, I have a question. In the previous study, what was the validation that the scientists were measuring microRNA that were exosomal? I didn't see that. Or were they just measuring microRNA in the circulation? Because microRNA can be within an exosome and it can also be transported with a carrier protein. And those microRNA would have different functions, one would hypothesize.
In most of these experiments that I demonstrated in this literature survey, it's not much is discussed about the transport in the exosome. It's just the idea that a microRNA can clearly influence the immune system. It's the transport thing is not studied in vivo experiments in a clear way. That is the interesting thing for the neuroimmunomodulation.
Right.
You want to have the release somewhere in the brain, the transport, and then the function in one single demonstration. And that is, I think, that's what we mean with neuroimmunomodulation, right? You would agree?
Yeah, and I do like the concept that, you know, the pesky blood-brain barrier is always a problem when we start talking about things moving from the brain into the periphery. But here in the past study that you chose to highlight, you have a peripheral sensory nerve, a neuron in the periphery, and that is a very interesting idea. So I wonder, for example, I'll have to do some digging to see how much of the peripheral nerve release, or maybe myelin, for example, a myelin sheath in some of these bigger peripheral neurons, what kind of exosomal release do those cells do?
I don't know, but that's exactly the question. How can it really be translated and from the brain to the periphery? These are certainly not simple experiments, but they would really show the links between the two systems. And that's
well, you know, a lot of the work with brain exosomes are done in a pathological situation. So you've got neuropathology on board already, which already means you've got compromised blood-brain barrier function. So it's possible that those are the cases, those are the examples where that kind of neural immune peripheral, like from the brain to the periphery, would be more prevalent than under a normal healthy condition. That's another interesting question. But
But I think the exosomes, if you have exosomes and you inject the exosomes, they can really appear in the brain. It's not an absolute barrier, the blood-brain barrier. And I think that has been shown. It has been shown that this can pass over the blood-brain barrier. You know all about these more open areas of the blood-brain barrier. There, the area postrema, for example, where the links can be, like we have it with the cytokines, like we have it with other substances. It should be demonstrated in a similar way.
Yeah, it's very interesting work.
Is it easy to tell from what type of cell is an exosome coming from? Like, is this something one can really say, oh, this is really coming from the brain, or the
Yeah.
other way around when
Well,
you collect it?
Teresa, that's a great question, or whomever asked it. And that's one of the big challenges in the field right now. And papers are coming out every day. It will require a protein that's unique to a cell or several, right? And this is always the challenge. You know, you've got some things that we know we can use, for example, with immunohistochemistry in the brain to know that this cell is an astrocyte and this one is a neuron and this one is a microglia. But it's a tricky business, and that is a big push in this field, is to look for cellular signatures that you can be quite confident this is the signature from a kidney cell, or this is a signature from a hepatocyte in the liver. This is a signature from a neuron, a peripheral neuron, right? And that's really exciting work. And I
Yes.
hope, yeah, you know, it could be really, really cool
Yeah,
because you
and
could,
that signature, Moni, that signature could be a fluorescent labeled thing, and then you would also find it in the tissue. You would
yeah,
find it if it would be a fluorescent labeled protein somehow, and you go into the brain, and then you would see under certain conditions. Let's say we have a stress, then
yeah,
this labeled thing might be found in the hypothalamus, might be found in the amygdala, and
yeah.
we might even see the travel.
Yeah, and you know, there's a challenge with injecting exosomes. Those experiments are actually very difficult to do in vivo because you're injecting these little vesicles that like to just go the places things that are injected go, and you end up with a bunch of them in the lungs and then a
Yeah,
bunch of
but
them.
I'm
You can't,
thinking of animals who have
right.
a
That's what. Right.
certain transgene that is
Yes.
fluorescent.
Precisely. That's the way to do it. That's what I was going to follow up with. That's the way to do it, is to do, you know, one of these transgenic animal models where you can get, a particular protein where you can put in the gene so that when that protein is upregulated, you can see it glow. That's a very cute way to do it. And I think that will also be right down the way. I don't like
And
working with transgenic mice just because I don't like mice, but I'm sure somebody will be working with them.
perfect, yes, exactly. I think we are
Yeah.
the science is so far that it could easily mark uh, an exosome. I'm sure.
Yeah, just a follow-up question on that, as one of the objectives of the webinar is also to bring awareness that the topic is welcome to the journal. Rainer, how important is the experiment in vitro for you?
Yeah,
Is this something that you will still willing to consider when the methodology is using in vitro models, or
I,
yeah,
in vitro is the platform of most microRNA research, and it helps in a certain way. But if you want to really demonstrate neuroimmunomodulation, you need the in vivo situation. It would not be possible to do that in vitro. And with my example that I showed from this Chinese group in brain behavior and immunity, they nicely did it in vivo, and all these experiments that really show the links connections must go in vivo. They can of course use in vitro aspect that has also been done in this paper, particularly when it comes to the end organ and the function in the end organ. It might be interesting to take just cells from the end organ and look how the microRNA works in this cell, and then the in vitro aspect is important. But the travel in a way must be demonstrated, and that's in vivo.
yeah. Thank you so much, Rainer. We are coming to the end of the session, and I really wanted to thank you both for these two wonderful presentations. I myself, as a layperson on the topic, have learned tons, and I hope the audience have also learned a lot. I just wanted to also thank everyone who joined us, and that's to remind that the recording will be available at the Research Viewpoints blog. We will also send it to everyone who have registered to this session as well. And yes, thank you so much for everything. And yeah, have a lovely evening, Monica. Have a lovely day, Rainer. And yeah, happy day to everyone. Thank
Thank
you.
you. Thank you, everybody.
Bye-bye.
Thank you.